Welcome to Proving the Point, where we discuss important research papers that you should know about. Today, we're going to talk about this paper called Soybean Isoflavones Prevent Atrazine-Induced Neurodegenerative Damage by Inducing Autophagy. So just to explain the title, isoflavones are a class of nutrient found in a lot of plant-based foods, including, of course, soybeans. And they've been shown to have many positive health effects, which we'll talk about. And what this paper shows is that these isoflavones can protect against atrazine-induced neurodegenerative damage. And what that means is there's this chemical, which is an herbicide called atrazine. You might have heard of it. And it has many negative health effects. So atrazine is sprayed on crops like corn and soybeans in order to kill weeds and thus increase crop yields and food supply and revenue. But it has many negative health effects, including, as this study shows and previous studies have showed, damage to neurons, which are the cells of your brain. So the idea is that this herbicide, we get exposed to it through the water supply, so it leaks into our water supply and we drink it. And also it gets on food residue. So a lot of pesticides, they're sprayed on crops. And then, of course, we later eat the crops. And even though they go through extensive washing, it's still thought, and studies have shown, that there can be pesticide residue that make it into the food that we eat then. So both through the water supply and potentially through food, people are likely exposed to atrazine, especially as we'll talk about in farming areas. Okay, so the idea is we get exposed to this pesticide, and then that causes damage to our neurons, and obviously that's a problem, but they go on to show here that there's this nutrient, isoflavones, that can protect against that. So that's what this study is showing. Okay, so before we get too far into it, why does this paper matter? Well, it matters because, one, it shows that there's a lot of pesticides that are harmful to our health. So in general, people don't really appreciate the fact that there are toxins literally everywhere in our environment, including on our food, in our air, in our water. It's truly impossible to escape them. So when you hear people talking about toxins in our food and stuff like that, there's still a group of people who view that as fringe science, but it's important to realize that it's not at all a fringe belief. It's incredibly well substantiated by mountains of evidence, which we'll talk about some of that today. But atrazine is but one example of an artificial toxin that we intentionally put out in our environment for a given purpose, but then it has negative effects on our health. And we're going to talk about the story of atrazine. Scientists have denied its harmful effects for a long time, and this has been around for almost 75 years, this chemical. And only now are people really starting to acknowledge, okay, it does have serious negative effects that need to be addressed. So the reason the paper matters is because it shows, one, science is quick to make mistakes. So when this chemical was first released, like I said, it's been many decades since it was first released, and only now are people really starting to recognize its many negative effects. Two, this paper shows that there are a lot of beneficial compounds in our food, specifically natural foods, like plant-based foods. So again, there are still people who think alternative medicine that prioritizes plant-based foods and natural foods and whole foods and things like that. There's still a group of people who think that's fringe science, all right? But in reality, there are thousands of chemicals in plant-based foods that have positive effects on our biology, and they're still being uncovered and still being learned about. So this paper highlights both the fact that our artificial environment is very toxic in many ways, and that the natural environment is very beneficial in many ways. Okay, which for some reason still seems to be controversial for people. All right. Another reason this paper matters is because it highlights both, again, how slow science can be to correct its mistakes, and it highlights an example of scientific corruption. So we'll talk about how the negative health effects of atrazine were actually covered up by the company that produces it. So again, there is a conspiracy involved in this. Okay, so let's get into the paper. So they start off by talking about how this herbicide, it's known to have negative effects on dopaminergic neurons. Okay, again, neurons are the cells of the brain. Dopaminergic neurons are specifically neurons involved in controlling things like movement and reward and things like that. It doesn't really matter too much for us. What we need to know is that this neuron system, dopaminergic neurons, they're often damaged in neurodegenerative diseases like Parkinson's. Okay, so when these neurons get damaged, that causes diseases like Parkinson's disease, which causes memory loss and other neurological problems. Okay, what this study is aiming to do, again, basically what they do is they take rats and they expose them to atrazine, this herbicide, and then they take another group and expose them to atrazine, but then also expose them to these isoflavones from soybeans to see if the isoflavones can protect against damage done by atrazine. And long story short, that is indeed what they find. So when you give them these isoflavones, which they abbreviate SI, that protects against atrazine-mediated damage to these neurons. Okay, so let's go through the introduction because there's a lot of valuable information in here. But again, atrazine, just to give some background, it was first created back in 1957, and then it was pretty much immediately released for commercial use in 1958. So that only 1 year span shows you just how little safety research could be done. Because again, science takes a long time. And the fact that it went from just being created to being put out for commercial use in such a short time window shows you how little safety testing could have been done. And that's a problem because, again, we're 70 years later, and the health effects are just now starting to be discovered. Then they go on to talk about how atrazine can be found in groundwater sources and surface water sources. So it runs off from fields where it's sprayed, and it can get into the water supply. And they cite some studies that talk about how people have been shown to be exposed to this herbicide. Okay, and how it can be detected in urine from farm workers and other people living in the area, which signifies that it is indeed getting into the body. So we are exposed to atrazine. And then they talk about how if you're exposed to atrazine for a long enough period of time, it basically accumulates in your tissues. So even if you're only exposed to a low amount at a given time, it could potentially accumulate over time to the point where it's actually a serious amount. And that can damage your tissues, including, as they say, causing reproductive issues, immune issues, issues with the central nervous system. And they go on to talk about the effects on the brain more specifically, which we're not going to get into too many of the fine details of like these different neural pathways that are affected. But just know that it's involved in regions of the brain that influence reward and movement and are relevant to Parkinson's disease. Okay, so they give some more evidence about how atrazine damages dopaminergic neurons. So DA, that's the abbreviation for dopamine, which is a neurotransmitter that is involved in reward and movement. And they talk about how damage to these neurons and neurotransmitter systems are one of the major things that causes Parkinson's disease. So it might be the case that atrazine is causing Parkinson's disease. So another reason that this paper is important is because in our society, we tend to act like disease, especially chronic disease, are just random, that they're just happening without a defined cause. And then we blame our inherent biology or, nature's flaws for why we're getting diseases, when in reality, a significant portion of chronic diseases are caused by human factors, like putting toxins in our environment. Okay, so it shifts the blame away from nature and back toward humans. Okay, so given that atrazine seems to be damaging the brain and potentially causing Parkinson's disease, they talk about how there's obviously a need to prevent that. They talk about how drug interventions have been shown to be able to protect neurons, but they have serious side effects. So we need to find something without side effects. And so then they switch to talking about how there's a lot of nutrients in natural foods that seem to protect against chronic diseases. So they talk about certain polyphenols, and these are a class of nutrients that have been shown to protect against Alzheimer's disease, which is also a neurodegenerative disease. They give other examples talking about how they can protect against inflammation and cancer and cell death and things like that. So again, plant-based foods, they're rich with nutrients. People don't really appreciate the fact that when you eat something like a piece of fruit, there are thousands, if not tens of thousands of different types of molecules in there. And a lot of them have health effects that haven't even been characterized by scientists yet. But we can be sure that it's going to be positive for our health. Okay, so natural food is better. It's our natural medicine. Okay, they talk then about how the way that these nutrients tend to benefit our neurons is by causing something called autophagy, which we'll talk about more later. But autophagy, that means self-eating. And what happens there is your cells basically eat away at the damaged parts of themselves and recycle that material to repair themselves. So they're basically purging themselves of anything damaged or unwanted. And the argument from this paper is that these nutrients, like the isoflavones from the soybeans that we're going to talk about, the way that they benefit our health and our neurons is by stimulating this process of autophagy, which causes our neurons to basically purify themselves of any damage or toxins. Okay, so that's what they are investigating in this study. Okay, so a couple things to say before we move on to the actual study results. One, they talked about a lot of the negative health effects in the introduction of atrazine, but it's important to note that there are many other negative health effects of atrazine. So again, you could just go to Google Scholar and type in atrazine and endocrine disruption, because this is a big one, as we'll talk about. Okay, and you can see that there are hundreds, if not thousands, of papers talking about the effects of atrazine on endocrine disruption, especially in amphibians. Okay, you could do sperm count in humans, for example. Okay, looks like there are some papers that have looked at that in mammals, so it might be affecting reproduction in humans. Atrazine and cardiomyopathy. Okay, that's like heart disease. Promotes death of cardiac cells. Okay, so you can pick your health outcome. Let's do kidney disease. People have done the research. There's plenty of research talking about the negative health effects of atrazine. And that brings up the second important point. You might be wondering if going all the way back to the early 2000s and a little bit before that, we have evidence that atrazine is harmful to our health. Why is it still being used? In other words, why have regulatory agencies not corrected this issue of atrazine-induced health problems? Okay, and the thing about these safety organizations is, like, for example, the EPA or the FDA or things like that, is while they're ostensibly meant to protect the population from like corrupt companies and dangerous compounds, oftentimes they do the opposite, which is they work with the companies that are producing those compounds to ensure that they can remain on the market even if there's evidence that they're harmful. Okay, so demonstrating that point is this other paper, which we're not going to go too much into this paper, but it's very interesting and worth a read. But it basically talks about the history of atrazine and the history of the controversy around it. So it says that the controversy surrounding atrazine involves the effects on wildlife, particularly amphibians, and the questionable decision-making of members of industry, government, and the legal system and institutions of higher education. In other words, industry, government, institutions of higher education, they're all cooperating to basically bend science to fit their economic, political, or ideological ends. And they go into that in detail in this paper covering the history of this controversy. But it involved the company that produces atrazine basically producing a smear campaign of scientists who are researching its health consequences, and they spent millions of dollars doing that. Okay, so if we scroll down to this table, they give a timeline of events. So again, atrazine, it was first produced in the 1950s, and it wasn't until, decades later in the 90s when a controversy started to arise. So The company that produced atrazine was Syngenta, and they hired this scientist, this Hayes, to basically assess the health effects of atrazine. He started to find negative health effects, but they obviously didn't like that, Syngenta. So he ended up quitting and said that they were trying to buy his silence. So he was going to publish results critical of atrazine. The company was trying to prevent him from doing that, allegedly. Okay. Hayes then complained that Syngenta was trying to prevent him from becoming tenured at his university, that they were trying to kick him out of like scientific organizations. And there's a variety of other like unethical practices involved here, including groups that were funded by that company, Syngenta, basically petitioning the EPA to ignore the relevant studies on its health consequences. In other words, the EPA is supposed to protect us from toxic chemicals like atrazine. Well, according to this paper, the company that produced atrazine, they basically petitioned the EPA to ignore the data and continue to allow atrazine to be used as an herbicide. Okay? And so that, dispute continued for a few more decades. More papers are being published showing that Americans are being exposed in drinking water, talking about the negative health effects on amphibians and reproduction and ecological damage caused by atrazine. It's all being covered up by Syngenta at this point. Syngenta had to pay $105 million to settle a lawsuit that alleged that atrazine was found in the water supply. So basically, this company is getting in trouble for all of this. The EPA is not really doing anything. It's not until later, the mid-2010s, when the EPA really starts to criticize atrazine and oppose its use. Okay, so now they're starting to say that it is indeed dangerous. So again, this is just a good example of how regulatory agencies, they're not always looking out for our best interests, and oftentimes they're corrupt. They're sensitive to bribery. They're sensitive to manipulation by corporations. And it also exemplifies how, again, scientists, they might be quick to make mistakes, like introducing a toxic chemical into our environment. They're extremely slow to correct those mistakes. So it's now been decades. There have been hundreds of papers published on atrazine, yet it's still in use today, right? And it likely will continue to be used because you can't really put the genie back in the bottle, okay? We've become dependent on toxic pesticides to maintain the food supply. So once you start something like this, it's really difficult to undo it. All right, so let's get back to the main paper that we're focusing on. So that's all the background about atrazine and isoflavones and plant-based compounds. Now we're going to talk about what they did in this study. Okay, so in this study, they took a group of rats, as I mentioned, and they split them up into different groups. Some of the rats they exposed to atrazine. Other rats got exposed to basically nothing as a control group. Other rats were exposed to soybean isoflavones only. And then other groups were exposed to soybean isoflavones and atrazine to see if the soybean isoflavones would protect against the negative effects of atrazine. Okay, and so that's their basic groups that they had. And then after 45 days of exposure, then they would do behavioral analyses on these mice, as well as they would isolate the brains from these mice and look at them under the microscope and also do molecular analysis. So we're going to talk about the results from this now. Okay, so we're going to skip over most of figure 1 because it's basically showing no findings. But basically what this figure is looking at is, does atrazine impact like movement of these animals? And it really didn't. Okay, so we're going to talk about just panel A just to show you how these graphs are set up, because a lot of the subsequent graphs are set up in the same exact way. Okay, but the way that these graphs work is if you look at the x-axis, these are showing the results of the different groups of rats. So the control group, this was the group that was exposed to nothing. This ATR, That's the abbreviation for atrazine that they're using. So this group of rats was exposed to atrazine. Okay. The next bar is the group that was exposed to soybean isoflavones. The 100, that's referring to the amount that they were given. So in this group, they were given 100 milligrams of isoflavones per kilogram of rat. Okay, so they basically tested it at different doses. Okay, this next group over, they were given both atrazine and soybean isoflavones, but this group was given 10 milligrams of isoflavones per kilogram of rat. This next group over was given 50 milligrams of isoflavones per kilogram of rat. And this next group over was given, again, 100 milligrams of isoflavones per kilogram of rat. So they did a different dosage of the isoflavones to see if it would have a different effect. But again, this first figure, it's showing basically no effect of anything. Okay, so the atrazine at 45 days wasn't affecting their movement very much. Okay, if we scroll down to this next figure, again, this one's not overly impressive and important for us, but what this is showing is a microscope analysis of neurons within these rats' brains. Okay, so this first square, this first panel, that's their control rats. So again, they weren't exposed to anything. So this is what a normal neuron looks like under the microscope. This large circle, that's the nucleus, which is where the DNA is housed. Okay, they're pointing out various features like what a healthy mitochondrion looks like and what a healthy nuclear envelope looks like. But ultimately, it's kind of difficult to tell what's going on in these images, and you shouldn't put too much stock in a microscope image if there's no quantification associated with it, because it's easy to cherry pick images to show what you're trying to indicate. Okay, but the big thing from this figure is that in the atrazine exposed group, which is the second panel, they note that the nucleus is smaller. They note that there's swelling in some of these organelles within the cell. And the idea, or at least what they take away from this, is that it seems to be the case that this cell is dying. Again, I wouldn't put too much stock in the particular images just because it can be very difficult to tell what's going on in an image like this. But what the author suggests is that atrazine seems to be killing the cells as evidenced by this microscopy result. There's more evidence that atrazine is killing the cell later that's more reliable. Okay, so we're going to skip over most of that. The first big result that we're going to focus on is table 1. So this is where it starts to get interesting. In this table, they're basically showing that atrazine causes both redox stress and inflammation in the brains of these rats. So redox stress, again, without getting too technical, everyone knows you need antioxidants in your diet. Well, that's because antioxidants, they prevent redox stress. So that's what they do. Redox stress, that basically just means there's chemical damage happening to the components of your cell, if we're going to keep it simple. So the more redox stress, the worse for the cell. Okay? And in this table, going across the different columns, you have, again, the control group, which was exposed to nothing, the atrazine exposed group, soybean isoflavone exposed group, and then atrazine plus soybean isoflavones. And then in the different rows, these are their different metrics of redox stress and inflammation. So this MDA, that's basically a damaged lipid in the cell that's indicative of high redox stress. So the higher the levels of MDA, the worse for the cell. That's all you really need to know. So if you look at the control group, their MDA levels was 0.46. The atrazine exposed group, it was 0.7, which is significantly higher. So atrazine is causing significant damage to these neurons. Okay, and this asterisk here, that means it's statistically significant. Okay, so they did statistical analysis and found that indeed the atrazine exposed group was worse. Okay, but if you look over in this fourth column, if you expose them to atrazine and soybean isoflavones, we're back down to 0.55. So it's not a complete rescue. It's not a complete return to the control conditions, but it is significantly lower than the atrazine exposed group. And that's what this hashtag means. It's significantly lower than the atrazine exposed group. So the soybean isoflavones seem to be protecting the brain from redox stress. Okay, the next row down, that's glutathione, GASH, and that's basically the opposite. So this one increases with basically better handling of redox stress. In other words, to rephrase it, the lower the levels of GSH, the more redox stress the cell is under. So you see that under control conditions, it's at 0.54. When exposed to atrazine, it's dropped to 0.36. So again, that signifies more redox stress because they're depleting this antioxidant. So glutathione is a major antioxidant in the cell. Okay. When you expose them to atrazine, but then also give them the soybean isoflavones, you get a rescue. In other words, the glutathione levels are back up to where they normally should be for the most part. So those first two rows, they suggest that the soybean isoflavones are protecting against atrazine-induced redox stress. Okay. Again, it's worth stressing that redox stress is very harmful to the cell. So this can damage everything from your DNA, causes mutations, can promote cancer, promotes metabolic dysfunction, damages your proteins in your cell, causes a lot of problems. The fact that atrazine is inducing redox stress, that alone is a very important result. Okay. Next up, similarly, inflammation is increased in a lot of chronic diseases, and it's harmful to the cell. So it's a sign of damage. These two rows, TNF-alpha, that's tumor necrosis factor alpha, and this IL-6, that's interleukin-6, these are two proteins that exist in the body that promote inflammation. So the higher their levels, the more inflammation and therefore the more damage the brain has experienced. So if you look in the control conditions, it's at 189. But if you look at the atrazine-exposed conditions, it's at 263, so much higher levels of inflammation after exposure to atrazine. So atrazine is clearly damaging these brains. But then with the exposure to soybean isoflavones, it's back down a little bit. So there's a partial protection by these soybean isoflavones. Okay, so the point of this figure and the similar results were observed for interleukin-6, another indicator of inflammation. So the main takeaway from this table is that atrazine is promoting both redox stress, which is very bad for your cells, and inflammation, which is also, one, a sign of damage, and two, it's in and of itself bad for your cells. So atrazine is damaging the brain based on those metrics, and soybean isoflavones are protecting the brain from that. Okay, so it's a very important result. So with this next figure, figure 3, what they're showing in panel A is basically the levels of this particular protein, which abbreviated TH. That's tyrosine hydroxylase. That protein is involved in producing dopamine naturally in the body. So in other words, the higher the levels of this protein in the brain, the more dopamine production can happen. And remember, dopamine, that's the neurotransmitter involved in movement and reward and things like that. Okay? And whenever that neurotransmitter is not being produced properly, you get neurodegenerative diseases like Parkinson's. Okay? And the way that you interpret this image is basically the thickness and intensity of the band, so this dark black band, the more of tyrosine hydroxylase is present, and therefore the more dopamine is being produced. And so what you can see is that you got a pretty thick band in the control group. When they're exposed to atrazine, the band is much thinner. So there's less of this tyrosine hydroxylase and therefore less dopamine, signifying a problem with your dopaminergic system. Okay, but then if you give them atrazine, but then also give them soybean isoflavones, that protects and restores tyrosine hydroxylase levels. So again, the soybean isoflavones are protective, and they quantify that here. So again, you can look at the different bars correspond to the different groups, and tyrosine hydroxylase levels are dropping, but then they're restored with the isoflavones. Okay, panels B and D, that's showing the same thing, but in a different brain region. We don't really need to focus on that. Okay, but the main takeaway here is that atrazine seems to be messing up the dopamine system, which has important health consequences. Okay, next up, this figure, this is showing results related to apoptosis, and that means basically controlled cell death. So what they show here is that atrazine basically promotes cell death of these neurons in the brain. So this BAX, that's a protein that promotes cell death. BCL2, that's a protein that prevents cell death. And what they're showing is that compared to the controls, when you expose the rats to atrazine, that increases the abundance of the protein that promotes cell death, and it decreases the abundance of the protein that prevents cell death. Meaning, atrazine seems to be promoting cell death. Okay. If you add the soybean isoflavones, that sort of protects against this. So easier to see in this quantification down here. So this is just showing the same. It's showing the data from panel A, except for in bar graph form, which is easier to understand. Okay. So you can see this, again, this is Bax levels. This is the protein that promotes cell death. Well, it's higher when exposed to atrazine. So atrazine is causing cell death. But then when you add the soybean isoflavones, that lowers it and protects against cell death. Okay, again, this next panel in panel E, that's showing BCL2 levels. That's the one that prevents cell death. So atrazine reduces the one that prevents cell death, which would again promote cell death. It's like a double negative. Okay, you're losing an inhibitor of cell death, which promotes cell death. When you add in the soybean isoflavones, that corrects it. Okay, and again, they show the same thing in a different region of the brain. And we're going to skip the rest of this. The main point is simply that they showed that atrazine is promoting cell death and that the soybean isoflavones prevent that. Okay, the reason that matters is because whenever your cells in your brain die, of course, that causes cognitive dysfunction, memory impairment, and all kinds of neurological problems. And it predisposes you to a lot of different diseases. Okay, so obviously you don't want the cells in your brain to die. Okay, we're going to skip through most of the rest of the paper, but they try to explore the mechanism of how atrazine is causing cell death. And they find that it's disrupting this process called autophagy that we mentioned at the beginning. So again, we're not going to go through all of these different genes involved in this. These are different proteins involved in autophagy. But the main conclusion of these last few figures is that atrazine seems to be preventing autophagy. Remember, that's when the cell basically purges itself of any impurities and makes itself healthier. So atrazine is preventing that, therefore causing these cells to be full of toxins and damaged components, and that causes cell death. Whereas these soybean isoflavones, they seem to be basically stimulating autophagy, stimulating this purification process in our cells, thus allowing them to be healthier. Okay, so they show that throughout this figure. They show it again in more detail in this following figure, but again, we're not going to go into it too much. And then they show it even again in this figure. So this result is showing microscopy results. We're not going to go through it. It's basically showing the same thing. But that's the last figure of the paper. So the main takeaway from this paper, like I said, one, our environment is full of toxic compounds like atrazine. They get in our food, they get in our water, they get in our air. We ingest them, and they cause all kinds of health problems. Everything from neurodegenerative disease to pretty much any other chronic disease that you can imagine. So again, diseases, they're not random. They don't manifest at random. Our population is actually getting less healthy by a wide variety of metrics, which we can talk about in future videos. And a lot of this is due to toxins in our environment. So we need to improve our environmental conditions if we're going to be a healthy population. Okay, and then one way to improve our health as individuals is to eat more plant-based foods. And, I should say now, it's not just plant-based foods, but a lot of natural animal-based foods are also very good for you. Okay, but this study is focusing on plants. Again, you eat a piece of fruit, there are thousands of chemicals in there that are influencing your health in very specific ways. So you see, they're showing that this one specific molecule, well, this is a class of molecules, but they activate specific proteins in your cells that have specific molecular effects. Okay, so it's all very complicated, and there are thousands of chemicals in our foods having these effects. So maybe we'll talk about some other toxic pesticides in future videos, because again, it's truly a serious and ubiquitous problem. But thank you for listening.